Hi. Actually, today I want to present, a short part of my research life and my current research work. Today I want to talk about two agricultural insects. One is Oriental foot moth, which is Grapholita molesta, and another one is cotton seed bug, is Orchicarinus hyalinipenis. So currently I'm working with this cotton seed bug. But at first I want to share my experience with Oriental foot moth actually is just a little one part of my PhD work. Uh, that this Oriental foot moth actually is the pest of that stone fruit and the plum fruit like apple, peach, pear, nectarines, and apricot, everything from stone fruit and plum fruit. So at first I want to describe about the Oriental foot moth part, and then later I'll discuss about the cotton seed bugs one. Uh, this the biology. I just want to say, describe the biology of the Oriental foot moth. So this moth has like a four stages, as you know, that is the egg, larva, pupa, and adult. And larva has a 4 to 5 larval instar, and it takes 30 to 40 days from egg to adult stage. And the thing is that they have multiple generation per year, typically like 4 to 6, is depending on the climate. And they're overwintering actually as diaposing larva is actually the cocoon larva before the pupal stage, and they stayed in the bark or in the debris, and female lay eggs on the shoots or on the fruits, the fruit surfaces. And here you can see the distribution, the red mushrooms, the distribution of this paste is distributed USA, China, Japan, South Korea, New Zealand, Canada, and so on. But first, it was introduced in the USA from Japan in 1913, now found across all over a major growing region. This Oriental foot moth is presently limited a small center region of Washington, but climate change projections that predict that it accelerated the spread into major fruit production areas. Now, it's actually in some part of in the US, it's considered as a minor pest because they able to control this pest in a nice way. But still, you can find is a problematic in some areas. And the University of Illinois Extension state that this Oriental foot moth is now well established in Illinois, with 4 generations per year in central Illinois and 5 in southern regions. Here you can see that it has like two hosts. It's like a primary host and otherwise a secondary host. So what primary host is like peach, nectarines, plums, and apricots. This Grapholita molesta is a major pest of peach actually. Peach, nectarines, plums, and apple. In the early season, it happening, the flagging occurs when a larva tunnel um, into the new shoots. You can see the pictures in the right side. There's you can see the twig. In the early season, like in April, early April or late April to early May, so it's normally attached the twig, the shoots, and then later stage is attached to the fruit. But in the early season, it attacks peach, nectarines, plum, and apricots. And later season, it shifts their host to apple and pear. Normally, the empty holes are often hard to detect actually. They need such a tiny holes. And damage actually found after the cutting of the fruit, or you can see the frost. Uh, in the second picture in the right side, you can see some frost in there. So it, once they go inside, it's very difficult to detect, or is that uh, infested by this fruit, this pest or not. But after getting inside there, you can easily detect, okay, this is they infected with the Grapholita molesta. How they monitor actually. Normally in USA, a pheromone traps for adult male flight is used to detect the presence of this pest, and they used degree models to predict the egg hatch, and also they can detect through early season shoot flagging. As I mentioned that one in the right picture, you can see that one. So there's the indicator of the presence of this insect, this pest. So here, my first part come. So when I was working actually on two projects, one is the plant stack and entomopathogenic fungus against this insect. So I ran into a real problem actually. My OFM colony, actually I was working rearing that insect on artificial diet, but I wasn't getting, producing enough insect for my study actually. Uh, then I was getting some mold, and it's the diet became dried, and I was not getting enough insects to run my experiment actually. Then I thought that okay, don't waste time. Uh, let's try to evaluate other natural food source. Uh, then I started working. My this work is development and fecundity performance of Graphylita molesta on different immature fruits. So I select peach, plum, and apple to evaluate their host strains. Okay, which fruits actually is better for Graphylita molesta? Our research goal is to to evaluate development and reproduction of Graphylita molesta in immature fruit of peach, plum, and apple fruits. Here I choose peach, plum, and apple, which is collected on 25 May. Plum also collected on 25 May, and apple just collected 14 June. And you can see that for the development and survival rate immatures, I use that breeding dish and a square breeding dish for the when they becomes adult. And for the adult, when they become adult, I just provided that 10% sugar solution for them. And let's see here the result. So here you can see the boring rate. Boring rate is like when the Phosphonistel larva go inside the fruits. The boring rate is higher on peach and plum compared to apple, whereas the duration of the larval stage also very lower on peach and plum and compared to apple. And immature survival rate also higher on peach and plum than apple. And but here's the thing: lifetime fecundity is higher on plum and apple. It's quite interesting, right? Boarding rate is higher on peach. Larval duration is lower on peach. Survival rate also higher on peach, but fecundity is lower. So anyone can tell me like, okay, if I wants to choose two fruits here, the best one, which one I should choose? Any case, plum? Yes, plum is number one. Then which one is the next one? How you can determine? Okay, because larval development is faster in peach. Survival also is, good in peach, but fecundity is low. How we can determine the best food source for Gaphilita molesta? Any guess? But in overall, like how you can quantify, okay, this is the best one and this is not the best one. There is a way. It's called live table analysis. So next one, I did the live table analysis. So here I want to emphasize the intrinsic rate of increase. So intrinsic rate of increase is that the per female per day the growth rate. So it tells you that how fast, how quickly the growth rate is happening. So here, to analyze the intensity rate of increase, that is the RM value, you need the survival rate, you need the total duration time, and you need the fecundity. So all three categories, you need to analyze this intensity rate of increase. And here it tells you, okay, plum and apple is the better food source than peach. Because here it's very difficult to judge, right? So it's better to have a live table study, to evaluate, okay, which gonna happen. So once you have live table analysis, you can predict, okay, what is the status of my colony and how it can work, the better way. Also, you can use this data to for the management purposes also. If you have the live table data, and you can predict, okay, when it gonna uh, go in a good way or in a bad way. So here we know that, okay, that peach is not a good one, though it has higher survival rate. It come very fast. The larval development is very shorter, but the fecundity. So everything is actually important to uh, give a emphasize on, okay, which food is better. So only the other parameters is not enough to determine. So if we have like intensity rate of increase, and that it tells about like, okay, so this is best or is no is not a good one or a bad one. So you can easily quantify that kind of things. I also tried, the host preference of Graphylita molesta on these three fruits. I choose, used a multiple choice essay. I release the inset in the middle and check, okay, which they can prefer. And if you see the result, peach has higher first choice rate, is about like a 38%. And the final choice is also, higher on peach compared to plum and apple. So they like peach fruit actually. Though they cannot make good uh, progress in that peach fruit, but they like peach fruit. The volatiles, the imitate form, the peach fruit, they like that one compared to plum and apple. But through our live table study, we found that plum and apple is the better food source. So it is quite interesting, that one. So the thing is that the plum and apple support better OFM food growth, right? But as I told you before that one, that I'm doing this experiment because one reason I want to establish my colony. If you see here, this study, boarding rate is lower on plum, right? But as I told you, that intensity rate of increase, it shows that plum and apple is the better food source. So I was working, so I need to work on plum and apple to rear this insect because my goal is to rear that insect to perform other experiment. So for that one, I choose apple for that experiment because I wasn't getting that plum fruit in that time. So I was working on the apple fruit. So here it comes. So punching apple fruits increases survival of Grapholita molesta in laboratory rearing. Why I choose that one? If you see that boarding rate is lower in here, right? But it works once they goes inside, it works well. They have proper nutrition to get produce more eggs, and the fecundity, if you see, the fecundity is too higher in apple compared to peach. So there's something that we can maximize their potentiality. Like boarding rate is higher, so that means if they goes inside, maybe we can get a better food for them. So what I did, I punctured that one. So my next uh, work was how to get them inside into the fruit. So what I did, I punctured the apple fruit. like around 20 punchers around the apple fruit. And I use like the apple fruit whose diameter is 6.2 centimeter. And I did the puncture using that insect pin is 2.6 to 3.2 millimeter deep, not too much, just a little puncture. And there's a sister application I tried. And the same procedure I followed from the previous study, the same, beating dish and using square beating dish for the adult and everything. And same conditions, I followed that one. Now, if you see the result, there is no significant difference on their developmental time. Egg stage, no significant than punctured and unpunctured one. And larval period, also similar. Pre-pupal period, also similar. Pupal period, also similar. And whole immature developmental time, also similar. So there is no significant difference on the punctured and unpunctured one. And then we checked their pupal weight, is also similar. There's no difference between these two one. And if you see the fecundity, there's also good. So fecundity is like 238 and energy is 234. There's no significant difference. Their longevity, their ovipositional period, the pre-ovulation period, everything is same. Wait, there's that thing. Their survival rate is 70%, like almost 63%, and unpunctured one only 37%. Here's makes the difference. So when we puncture that one, they can easily go through that one and it increase their survival rate. Though every other parameter is the same, but overall survival rate is higher on punctured one compared compared to the unpunctured one. And there's a entomologist actually, Bisognin et al. 2012. He also evaluated apple fruit with artificial diet, and he found that normal apple fruit is overall survivability is 30%, but our one we found that unpunctured apple the overall survivability is 37%, but artificial diet it produced like 54% overall survival rate. But our study shows that if we puncture the apple, the survival rate increased to 63%. So puncturing apple fruit, it works. So from there, actually, I started the rearing the Oriental food moth using this puncture apple fruit. Uh, yes, I think maybe you have some question in your mind that okay, if we puncture, maybe it will, um, maybe it's not like works through what we expected. Like maybe it will get cold immediately. Get cold mold immediately, right? Because we are puncturing, we are hurting the apple. But the thing is, it's happened. It's not thing that is not happened. But they need only 12 days to, or 2 weeks maybe, is approximately maximum is 2 weeks. They can easily go through that one and they can easily come through from the fruit. So using the apple fruit is works very well. Uh, and there's another reason that why you choose the apple. Maybe we can use some other fruits, maybe. Um, but apple is, normally growers do pruning, and they need to throw the apple fruits, the immature one. So maybe we can use that one, to save some money for, making the artificial debt. The another good thing is that we are getting higher survival rate. So that work done very effectively. So from that time, I started rearing that insect using the puncturing apple fruit. Now, still there is a question. As I showed you in my first work, that boring rate is lower on apple and fecundity is higher on uh, apple, but intensity rate of increase also. That means what I wants to say that we found that plum and apple fruit is the better fruit source, right? Immature plum and apple. But lots of, scientists say different things. They say that peach fruit is better than apple and plum. But here's the thing. They tried on mature or ripening fruit. They didn't evaluate the uh, the immature fruit. So here's the question, like, okay, my next work comes out. Developmental stages of peach, plum, and apple fruit influence development and fecundity of Graphylita molesta. So here, I wanted to know what is the reason actually from there, like in immature peach, it was unsuitable, but later it stays suitable. What is the reason behind that one? That is still that time it is still on our that what is the main reason behind that thing? So here I just um try to get that answer, how we can find out, okay, when it shifted their choice from uh, peach to apple, or when it becomes more favorable for them. Here, then I evaluated different stages of peach, plum, and apple fruit. And what is the reason behind that one? As we know that sugar content is very essential for the larval development. So here we choose two assumptions. Maybe they have higher firmness on the fruit, or maybe is the sugar content also affected them to form unsuitable to suitable things. So we hypothesize that the firmness and sugar content may affect the development and reproduction of oriental foot moth. To test that firmness, we used that universal uh, testing machine. Is actually force displacement measurement. To test the firmness, is they can measure up to 500 Newton pressure. And to test the sugar content, we use the sucrose fractometer. Here you can see that when we collected the fruits, plum we collected on 11 May, 31st May, 11 June, 25 June. To evaluate that thing, we should get the fruits in the same time, right? And we didn't get peach and apple fruit on 11 May, but we got plum fruit on 11 May. So uh, uh, we analyze using that fruit on each time. So it's, it would be better if we get uh, peach fruit from 11 May, but we couldn't get that one. So here you can see some interesting result here. If you see in the firmness in the plum fruit, when was in May 11, the firmness is higher. Also, the sugar content also lower compared to the later stage. But that the pupil weight is quite good. It's not bad, like it's not lower one. It's quite good one. But the firmness is going down when the season is approaching, right? The next season. Then firmness decrease in May 31st. And sugar content also higher. So that time we get higher number of people weight, higher people weight. What happened next? In June 11, if you see, firmness is going more down and sugar content also higher. It's a good, right? It's a good scenario. There's no firmness, there's no hardness in there in the fruit. Sugar content also higher. So we should get good number of the progeny, or the people weight should higher. But no, it goes down. There's another interesting thing. It's not true that, okay, oh, is not farm, um, it can easily, the get the OFM can easily get the nutrition and everything, but it's not true. So they need a certain amount of the firmness and the sugar content. I want to describe like a peach one. If you see peach, the earliest is in May 31st is a higher firm. firmness happen in peach fruit, and sugar content also little bit higher, but we got very the people weight is very low. Uh, but when the time goes, when we collected the fruit on June 11, the firmness is little bit goes down and the sugar content also goes higher. Then we got the higher people weight. And the next stage in when we collect the fruit on June 25, again the firmness goes down and sugar content goes high, and we got a very good number uh, in people weight. The people weight is higher on peach and later stage. And if you see apple, it's also like the sugar content weight is goes up, and we are getting uh, higher people weight for apple. If you see that the duration of immature stages in plum, the duration is lower on the early stage, shorter, and higher in the later stage. And like in plum, if you see that in when collected on May 1st, May 11, and 31st May, there is no significant difference. But in the later stage, on 11 June and 25 June, the larval developmental time is higher than the previous one. We didn't get any difference on peach fruit. Every time same. But the trend is going down, but there is no significant difference on the duration of the larval stage. Same thing happened in apple. When earlier stage, the duration of the larval stage is shorter compared to the later stage. And if you see the larval survival rate in plum, the larval survival rate is higher on the early stage on 11 May and 31st May. Then it goes down. In 25 June, in June 25, the survivability is significantly lower than the earlier one. We didn't get any significant difference on peach fruit, but you can see that there's a higher trend in peach fruit. And in apple fruit, also there is no significant difference. So for the larval survival rate, that peach and apples is quite same. Um, and the total immature, actually, we didn't get any significant result because there's a people duration, there's uh, another parameters also there, but we didn't get the immature survival rate. There's no significant difference, but the larval survival rate is um, similar in peach and apple, but in plum, earlier is better than the later collection date. Now here you can see the fecundity. But in fecundity, if you see the plum fruit in the left figure, the earlier stage one in May 11, there is fecundity is similar with the next one, and that means the later season. But in 31st May, it becomes significant than the May 11. But later, it goes down actually. We didn't get enough eggs from the adult. But in peach, in earlier stage, is very low. But once it becomes, uh, you know, uh, bigger, and the later stage, we found that the fecundity is significantly higher than the earlier collection date. And there is no significant difference in apple. So every time is same in apple fruit. And if we compare them within the fruit species, like in if you see that in the lower one, the lifetime fecundity actually is significantly lower on peach compared to plum and apple. That's what we got, no, in our first conversation. And then love in June, what happened that fecundity goes down that is compared to actually to peach and apple is actually lower than peach and apple. And 25 June, it goes more down and is significantly lower than peach and apple fruit. So, as I told you, that it's very difficult to quantify that one. Okay, which one is the better one? So we did again live table analysis and found that actually in earlier stage, plum fruit is better. In later stage, peach fruit is better. And apple actually earlier is also better compared to the later one. But what we found here, plum fruit earlier is better and peach fruit later is better. That's what everyone is saying, right? That mature peach fruit is the better food source for oriental foot moth, not the immature one. So we know that, okay, now what is the reason actually is going behind that one? Because we found that the intensity rate of increase actually is higher on uh, peach fruit in later stage. Because if you see the right picture again, I'm showing you that one, that in later stage, the firmness is lower than the earlier stage in peach fruit. So that's why, and the sugar content also going higher. So that's why probably is the reason for getting more number of eggs. But here's the another thing. There's a question that, okay, though we are getting enough number, but the survival rate is still lower. Why this has happened? Because as I say that, okay, there's a more less farm, higher sugar content, but there is still not able to enter into the fruit. It's not only about the firmness and sugar content, it's also the pubescence. That means the hair. Scientists found that actually the hair on the peach fruit is acts as a deterrent for the insects. So there's the another reason they like that one, they like that fruit, but couldn't make enter into the fruit because of the dense hair, or you can say the pubescence. So there's the another reason actually we are not getting enough insect higher survival rate for that insect. So here we know that okay, in apple, highest level of food penetration, moth reproduction, and overall RM were observed, probably due to apple have proper hardness and a higher sugar content, and plum fruit showing a higher larval penetration rate, were generally unsuitable for development and reproduction in the later stage because of the rapid liquefaction. So, liquefaction. So the thing is that though plum fruit is better, but the later stage they are not a good food source because it's liquidified very easily once they get inside. So they are not getting enough nutrition from the plant food. So that's why the later stage plant food is not better. So we measure actually which amount is needed uh, in this in this work. So we found that hardness, that is the firmness, ranging from 7.86 to 9.74 Newton for firmness, and sugar content range 8.85 to 10.98 Brix is the better time they can produce more, or they can be a good food source for Grapholita molesta. So we know the reason that okay, when is shift their unsuitability to suitable one? What is the reason? There may be some other reasons like, okay, maybe lipid profile or maybe a protein profile. We should get that one to quantify that result, but we couldn't do that thing. Maybe next time we'll be able to get that kind of information on different developmental stages, what is happening. So this is the small part of my research career that this kind of I'm at first my intention was to find out okay rear that insects, but later I got different information and I did different research work. So there's a small part I just um, described that one, and this one I want to explain about my current research work. The thing is that I'm very sorry. Actually, I didn't get approval to present all my data uh, uh, from my institution, actually. Currently, I'm working on cotton seed volatiles, actually functioning as host-seeking cues for cotton seed bug. Uh, and you can see that currently is becoming a major pest of cotton. So here, what we want to do, like to get uh, the pheromone or the pheromone actually, because there is no monitoring tools to detect that insect. So we are trying to develop one pheromone, aggregation pheromone or the pheromone to detect that insect, and we can use it in the field. So our lab actually focus on chemical communication and insect behavior. What we do, we evaluate semiochemicals, that is allelochemicals and pheromones. Allelochemicals also has like chiromones, allelomones, and cinnamones, and pheromones. And currently, I'm working on chiromone and pheromone, like aggregation pheromones, sex pheromone, and alarm pheromone. And the thing is that uh, uh, we're not supposed to tell you that what kind of chemicals actually we're getting from here. So but I can tell you that our lab actually focus on understanding and disrupting chemical communication system of insect pest and other arthropods. Goal is to develop effective lures or repellents and findings the real world pest control programs and transfer the knowledge uh, to the pest for the insect IPM technology. So here, I'm currently working on the chiramone one, and chiramone is a semiochemical that released by an organism that benefits the receiver, and while potentially causing harm or no benefit to the emitter. And our objective is to analyze an evaluation of the volatile compounds from cotton seed that are responsible for cotton seed bug attraction. Here you can see the workflow. In phase one, we collected the cottonseed volatiles, collections and record chemicals, and sample preparation, and then we do a GCMS analysis to uh, analyze the compounds from the cotton seed. And in phase two, we collected fifth instant name to get that unmated and mated males and females to test on that one, and then simple sample preparation, and then we test it in Y tube olfactometer. And then the next stage, we make a natural blend using the individual compound which actually active for cottonseed bugs, and later we just use different other blends to test it that okay, is there any additional blend that can work for this cottonseed bugs. And you can see that we got actually total 17 volatile compounds. I cannot tell about the compounds actually what we're getting because of the issue with the patent. Actually we applied in the patent, so I was not able to tell you what kind of chemicals actually we got from there. So we got 17 volatile compounds, and here is the thing: we tested on males and females, on mated, unmated in males, and mated, unmated on females, the same way. And we want to check and how we did that one. So at first, we collected fifth instant name and keep them in the individual vial. So when they becomes adult, we test it accordingly. If we want to get female, unmated female, we got from there. We keep them individually. But if we want to get a mated one, we keep them together and get the mated one. And here you can see that the white tube procedure and how we did that one. And the observation time actually 5 minutes, and age of the CSB adult actually less than 10 days. And we keep the temperature for 25 degrees Celsius. So here you can see that there is the attractant and the control, and that cotton seed bugs choose the attractant. So here you can see at first I um, tested individual compounds, all 17 compounds, and found that these number 7, number 10, number 14, and 17, these four compounds actually uh, shows better efficacy compared to the other chemicals, both on males and females. So in the next stage, actually, we make the natural ratio. We got the natural ratio of these four compounds, and we test it in different doses using this four compound. And we found that this dose 0.01 is actually effective on both sex, actually males and females. We also tried different random blends using 1:1 ratio. Um, in with using this four compounds. In the figure A is a blend of four chemical compounds. It also works on the 0.01 microgram dose. In B, we take out one chemicals from them, and we found that there is no significant difference in any of the sexes. In C, we keep only just two compounds from there. We also didn't get anything from there. In D, another two component, we keep them there. We found that one microgram they showed good efficacy, but in E also we keep them three compound there, but we didn't get. But what is the thing is that uh, in both natural and um, that random blend, both actually works uh, on that 0.01 microgram dose using this four blend. So uh, there's the additional blend, as I mentioned, that blend four that is using two component from there. It's also showing in a higher dose, not in the lower dose. So we can say that actually both the natural ratio and randomly formulated blends containing all four components showed a strong behavioral response from both male and female CSB adult at a dose of 0.01 microgram. I'm very sorry, actually. I couldn't show you the results and the chemicals, uh, but I can tell you what we already done that one. We already identified the pheromones, the aggregation pheromone, the alarm pheromone, and we did lots of field work. We found that they choose a non-host plant, and we find out the result also that, okay, why they choose that one and what is happening in there.